Our group conducts creative and innovative research with the aim of ensuring sustainable use of aquatic resources, protecting healthy ecosystems, reducing poverty and hunger, and developing capacity in these areas. The two young scientists who will present today are outstanding ambassadors for our group, who have taken their research from Africa to the world. We are also particularly proud that they have chosen to continue their careers here at SAIAB. We very much hope you enjoy their presentations and hearing about their journeys. If you have any questions, we invite you to leave them in the Q&A section on Zoom, and we will answer all the questions at the end of both presentations. Our first speaker today is Nobuhle Mpanza, a first-year PhD student at Rhodes University based at the South African Institute for Aquatic Biodiversity. She identifies as a marine biologist, but you can always call on her if you need any work done in freshwater ecology and biology, specifically working with invasive species. Apart from her science career, she volunteers as a peer educator and Oppidan mentor for first-year students at Rhodes University, which she says is her way of giving back and uplifting others in her community. I'm very pleased to introduce Nobuhle Mpanza.
Thank you, Josie. Good morning, everyone. I've been introduced, so I'll just get straight to it. So before I get into my research, the first thing that I wanted to share with everyone is where it all started. I started my undergraduate degree in marine biology from the University of KwaZulu-Natal, of which immediately after finishing, I got this amazing opportunity to relocate from KwaZulu-Natal to the Western Cape. And then where I did my honors in biodiversity and conservation biology, still specialized in marine biology. As it has been said, I am a marine biologist. But when the opportunity came for me to do something in freshwater ecology, specifically looking at invasion biology, I took the opportunity because I'm always willing to learn and adapt. So that's when I once again relocated from the Western Cape to the Eastern Cape, which is where I'm currently based now in Makhanda. And this is where I started working on freshwater, as well as the fish that live in this ecosystem, specifically invasion biology. So I keep talking about invasion biology and freshwater ecosystems. There's a number of issues that freshwater environments are facing, but the most prominent one and problematic one is the introduction of invasive species. So you might be asking, what are invasive species? So invasive species are fish or plants, animals or plants that are translocated from their place of origin into new places where they will not naturally occur. And why are they introduced into these new places? Mainly, in the case of the species that I'm working on, it was introduced in South Africa for aquaculture, but other species can also be introduced for sport fishing, pot trade, etc. So specifically, when looking at Nile tilapia, which is scientifically known as Orychromis niloticus in South Africa, it is a conflict species. So firstly, there is management of invasive species in South Africa, but it's kind of complicated by species called conflict species. Conflict species, my Nile tilapia, the invasive one, is a conflict species. And these are species that, as much as we recognize the ecological impacts that they have, as much as we recognize the ecological impacts that they have on ecosystems, they're still important. There's still this economical value of these species because they're being used for food security as well as in the reduction of poverty through agriculture. So my two species is Nile tilapia, which is invasive in South Africa, as well as Mozambique tilapia, which is native in South Africa. So Nile tilapia was first introduced in Limpopo for aquaculture, and then it's escaped from these aquaculture farms into nearby river systems. And now we're finding it where it was initially introduced for aquaculture, but then we're also finding it in some parts of KwaZulu-Natal as well as the Western Cape. And then within the Limpopo and KwaZulu-Natal, the Eastern Cape areas, we're finding Mozambique tilapia as a native species in these areas. But then there's also some extra limiter range where we're finding it in the Eastern Cape as well as in the Western Cape. So why then is Nile tilapia such a successful invader? So there are just a few attributes which makes Nile tilapia an invasive species successful in terms of invasion. Firstly, it spawns multiple broods within a single season. It has high parental care. It attains sexual maturity rapidly. But also, when you compare it to the native Mozambique tilapia, it has a broader diet compared to that one. Another important factor is when it comes to the climatic suitability of these two species. Nile tilapia, the invasive species, has a broader climatic suitability compared to the native Mozambique tilapia. This is very important, and I'll highlight as I go by. So how are these two species linked? There's a lot of literature published in terms of the trophic and habitat overlap, as well as the hybridization between these two species. And I just want to highlight something on the hybridization. So currently, in terms of NEMA in South Africa, the native Mozambique tilapia is currently categorized as a vulnerable species, which is at the risk of becoming extinct. And this is mainly because of the hybridization between Mozambique tilapia, the native fish, the native fish with the invasive one. So we're at the risk of losing the genetic pool of Mozambique tilapia because of this hybridization. And this is where my interest comes in, not specifically looking at hybridization, but I am looking at functional responses, where I want to understand the feeding dynamics between the two species. Firstly, looking at is the invasive Nile tilapia eating more than the native species. But also I wanted to understand if the invasive affects the feeding of the native. So I did my functional responses where I kept them in these big tanks, held them into these big tanks, and the prey that I used for my experiments, was chironomids. And then the small tanks where I've written experiments is where I was holding them for 2 hours to feed. And the experimental setup, I had a single species, but then Nile tilapia was allowed to feed alone. Mozambique tilapia was also allowed to feed alone, which was a single predator experiment. But I also had situations where they're conspecific. Nile tilapia will be eating with another Nile tilapia, or Mozambique tilapia will be eating with Mozambique tilapia. And then I had heterospecific situations where the Nile and Mozambique tilapia will be in the same experimental container feeding together. And for the sake, in the case of the heterospecific, I had to cut them open to understand who has eaten how much. Because as much as you can count what's left in the buckets, if it's just a single species, it's easier to do that. But when they're together, as much as you can count what is left in the container, you're actually not sure who has eaten how much. So the verification through the gut contents made things easier for me to understand who has eaten how much between the two. That was the first part of my MSc of the 3 chapters I had. And then the second part, I looked at their morphological differences. I measured a various number of morphological traits where I wanted to understand if there's differences in morphology between the two species, but at the same time, I wanted to understand if there are differences. Are these differences placing are they allowing the invasive species to use or to eat more than the native Mozambique tilapia? So those are my results for the first part, which was functional responses, looking at single predator as well as heterospecific, with the inclusion of the gut contents. So for situations, the orange is Nile tilapia, the invasive species, and the green is Mozambique tilapia. So in both situations, we found that the invasive Nile tilapia was consuming more prey compared to the native Mozambique tilapia. And in both cases, Nile tilapia had higher attack rates in comparison to the native one. And then the second part of the functional responses was looking at multiple predator effects. Also, whether it was observed or predicted, still it was the same case where we were finding that the invasive species is eating more prey than the native species. And then when I'm looking at the second part, which is the morphology, I found that, of course, there are differences between the two species. And these differences are allowing, when translated to the trophic profiles, these differences that the species exist between these two species are translating to the invasive Nile tilapia eating a diverse suite of aquatic food resources when you're comparing it to the native one. So there were items that Nile tilapia would eat, and you'd find that predictive that the native species was not eating. So what does this all mean in just a summary? So the greater take rates and the invasive Nile tilapia means that the invasive Nile tilapia is more successful in finding food items, even if they are available at very low densities. Secondly, the higher functional responses in Nile tilapia, the invasive species, means Nile tilapia has a greater predatory potential and also has the potential to outcompete the native Mozambique tilapia. And also the fact that there was Nile tilapia did not really impact the feeding of the native, but also the Mozambique tilapia did not offer any biotic resistance in the feeding of the invasive. So it continued to eat as normal as if there was no competitor in the system. And then the morphology parts in terms of the dietary overlap also is evidence of exploitative competition between the two species. And when it comes to, when I was translating the morphology into the trophic profiles, when it comes to the invasive Nile tilapia eating a diverse suite of food items, that means that the invasive Nile tilapia can withstand and overcome competition for food resources in the case that food items are becoming scarce in the system. So there was a lot of science. So now I'll move to the exciting part. At least it's exciting to me. So the research that I just summarized to you guys, I got this amazing opportunity earlier this year to travel to Belgium and present my work at the ICAS conference, which is a conference on aquatic invasive species. So this was exciting for me. I've never been outside of Africa. So this was my first out of Africa experience. Firstly, I'd like to thank the ICAS because I was awarded a bursary to travel to Belgium, but also the South African Institute of Aquatic Biodiversity, which is where I'm based, also afforded me the opportunity by giving me funds, travel insurance, and all those things to make it possible for me to travel. So on the conference, the exciting stuff about the conference is, firstly, presenting at a global stage is amazing, especially for me as a young scientist. But secondly, one thing that happened there was the morphology chapter. I've been reading about Leo and his work, of which the morphology chapter was literally based on the work that he has done. So when I was at the conference on the session that I was presenting on, he was presenting before me, like earlier on, and I was presenting in the afternoon at the same session. When I was presenting, he was sitting there listening to my talk, which was scary, but at the same time, very exciting. And I don't know if you can see, but there's like a picture on my far left. That's me after my talk, and Leo stood up, asked me a few questions, of course, about his model that I used. And then that was one of the highlights for me on the trip. But also after the conference, after the session that we had, he was kind enough to actually take me outside. We sat together, we talked. He wrote his email for me, which is exciting because it was like, if you need collaboration or anything, just know I'm available. And that was exciting for me because as a young researcher who's using someone's model and you finally meet this person face to face, that was amazing. It didn't end on just the science. It didn't end on just the scientists presenting their work. There was a lot of networking. We're meeting people from all over the globe. I think there's a group from America and Germany, and there's some people from Canada. And then another exciting thing that happened for early career researchers like myself is one of the nights we had a quiz evening where we're learning all about invasive species in Europe, but also learning about Belgium and the Netherlands since they were the hosting countries, which was exciting. I got a lot of contacts and emails and numbers. Yeah. And then the next part was the exploring, of course. If you're in a new country, you need to explore. So since Belgium and the Netherlands again were the hosting countries, we got to travel to the Netherlands. We drove from Belgium to the Netherlands, and then we had spent an entire day there where we were exploring the different cities. We went on a ferry. We went to identify some invasive species. We also scavenged for some oysters, and people were eating them there. So yeah, but yeah, it was exciting. But also, apparently, people say if you're in Belgium, you need to have waffles or chocolates. So this is me with my small bag after getting chocolates, which I brought home, of course, and that was also exciting. But I wouldn't have had the opportunity that I had if it wasn't for these amazing people. Firstly, I'd like to thank Prof. Olaf, who is the initiator of my whole MSc project. I came this side to Grahamstown because I was going to be working under him, and he had sold me this amazing projects that you're going to be working on together. So thank you to him, and also the opportunity to go abroad, but also in honor of Prof. Olaf, because I presented in a session that was honoring his name. Secondly, I'd like to thank my two supervisor, Prof. Brian Wasserman, as well as Dr. Josephine Pegg, who have carried me throughout my MSc base camp. Another exciting thing, there's a lot of exciting things. Another exciting thing is Ross, who is a co-author on my functional response and morphology paper. So I've been working with Ross a lot on the analysis of functional responses last year, the whole of last year, working together through emails, which was the only communication we had. But then when I went to Belgium, I had the nice opportunity of actually meeting him face to face and then talking about the work that you're doing together. And it was also amazing. So yeah, that's me. Thank you so much.
While we change slides, I'd just like to introduce our second speaker. Dinah Mukhari was born and raised in Mokopane, a small town in Limpopo. She studied for her BSc and honours at Wits University, and she joined Olaf Files Freshwater Group in 2020 for her MSc on invasive carp, which is the topic of today's presentation. She's currently a PhD candidate at Rhodes University in the Department of Ichthyology and Fisheries Science under the Acoustic Telemetry Array Platform team at SAIAB. Dinah is the elected student representative at the South African Network for Coastal and Oceanic Research, SANCOR. And we're very pleased for her to present her work today.
for presentation.
Good morning, everyone. As Josie has nicely introduced me, I won't spend much of the time introducing myself again. Today I'll be presenting on a chapter of my MSc, and this chapter actually is very dear to me as it was one of the ways in which Olaf sold this project to me, coming here in Makhanda in the first place. So yeah, tune in to hear what this is about. So starting with my study species, it's common carp. It's a fish that is very native to Central Europe and East Asia. And then I have a red circle around where its native area is. And then all the other circles around the world is where it's being introduced. So you may ask yourself, why is this fish moved all around the world? Especially, I mean, look at America, for example, and then all the way to Australia and South Africa there at the bottom of Africa. So this fish is introduced for multiple reasons, mainly food, and then as well as to develop people's livelihood, basically. So it's used as an aquaculture fish species, and then now it's also been introduced as a recreational angling fish. And then I've got a picture of the fish here. So this is the big picture is the adult, and then the small one there, which is almost the size of a match, is juvenile, the small form, yeah. And then, as I mentioned that this work is based on my master's, I did both the biology and the ecology of this fish species in Groenvlei. Although today I'll be focusing on the ecology, the movement of it. But then from my biology chapter, what I've learned is that that small fish that is the size of a match, which is the size of an adult in 2 to 3 years, and it's matured at that age. And then it stays around the same size all the way up to 20 years, which is amazing. And then I've also went on Google to find how long or how big this fish can get, and it says about 40 kg. And I'm not sure how true that is, but then I found this, which is the biggest. And then this was 25 kg though. And then, but I just wanted to show you how big it can get. I'm sure I couldn't carry that one myself. Yeah, going forward to introduce my study site, which is where I did my research. So Groenvlei is a lake, and then the Groen meaning green lake, and I'll tell you why it was called that. Well, why it is called that. So this lake is in Western Cape Province of South Africa, right at the bottom next to the coast. So if you look closely, you see that I have the Indian Ocean shown there, which is about 2 kilometers away from this lake. And in this lake, you also notice that it has no incoming water or outcoming water. So it's an isolated system that is maintained by rainfall and groundwater. So meaning that the species that are inside this lake are either introduced by people or have been here historically, which is about 5,000 years when this lake isolated itself from the ocean, which is a long time ago. So the lake is used by people from the locals, mostly for recreational angling because it has 5 non-native species that people introduced. One of them being largemouth bass. Here, one of them shown in the picture. And people love to go there to do angling specifically for that fish. And they will even host competitions to catch largemouth bass. And then other people do come to the lake for subsistence, where they just catch fish for eating at home and so on and so forth. And then there is us who go to the lake to do a lot of research on it. So yeah, that's the users of the lake basically. And then, okay, so this is a picture taken by a drone. This was in Groenvlei back in 2019 before I got here. So January 2019, this is how the lake looked. This is December, the same year And if you look closely, remember when I said this lake is called Groenvlei, which means green lake, and I said I'll get back to it. So what I meant was that they call it Groenvlei because they used to be able to see all the way to the bottom of the lake, which is full of reeds. So if you can see on the first picture, it's very clear. You can even see from the drone that this lake was very clear. You could see the bottom. But then with the introduction of carp in the system, now the lake looks more like, around the picture from December 2019. And here's why. So carp has been introduced all around the world, yes. And then it was a major food source back in the days. And then now in America and Australia, this species is called a Nuisian species. And this is because after a long time you have the species, the impacts are starting to appear. At first you wouldn't see them, but then after some time with the species in the water, impacts are starting to show. And these impacts are because carp feeds in a very particular way. It feeds from the bottom surface, and then when it eats, it disturbs the sediments underlying. It's an omnivorous, feeds on everything at the bottom, and then it takes everything in and then splits out everything it doesn't need, meaning that it's mixing everything at the bottom. So in doing so, it increases the turbidity, which is, as you can see, the milkiness. And now everything is now suspended and you can see through. This will affect a lot of processes, a lot of fishes as well, which use their eyes to feed because now there's no light penetrating, which means they'll struggle to see. And then other plants will also be uprooted in this process of feeding, which means microphytes or plants that live in the water will be dying most of the time. So what we did for this project, the ecology of carp, which is mostly movement behavior of carp, we started by catching this carp at Groenvlei. There's Hema there. This is carp in Groenvlei. I think this was our largest, which was about 5-point-something kilograms. And then we caught them and then put some tags in it. I have a picture of somebody holding the tags, and then you choose the size according to the species you're tagging. Put some tags inside the fish and then stitch them up. So a tag works almost like a tracker if you were to put a tracker in a person and see where they go. So now to know which fishes we tagged, we also put an external tag, which is shown at the bottom there on like a white line, which has our names and our contact details so that if you catch it by mistake or anything like that, you can call us. And then before we release them back, we make sure they are surviving and they are good to go back. So we put them in a recovery bucket for a little bit, look at them, watch them swim a little bit more before we release them back into the water. So now when we release them back, they have our tags, and then now we know that we're gonna track their movements. So this is a process called manual tracking. And then what I'm holding there is it's a VR 100, and then you put the pole that I put in the water there is the one that listens to the fish. So each fish that we tagged had a specific frequency in which the tag is releasing its ping. So we listen for the ping using the hydrophone, which is the thing that I have in water there. And then you drive the boat in a way that you follow where the fish is, and as soon as you come close to the fish, you take the GPS coordinates. And we did this for all day long and all night long. So I have pictures of us working during rain, during summer, during the night, and one of my favorite memories, now it's a favorite, but back then it was terrifying. Because when I was my first time in the lake at 12 o'clock midnight, I was very scared. I was wondering what am I going to see today, because I've heard stories about mermaid at Groenvlei. And this was one of the most terrifying day. And I was with Betty and Paul from Canada. And I don't want to tell them that I'm too scared, but I was just hoping that we make it tomorrow, which we did. Cool. So continuing with manual tracking, we went for 5 months, not 5 non-conservative months, because we were disturbed by COVID-19, and this is us in the lakes trying to make the best of it, because we'll be tracking for 24 hours. So within that 24-hour period, no sleeping. So we'll be trekking every 2 hours. So in those 2 hours where we rest, we'll be trying to look at other stuff. I'll be trying to entertain them. We'll be, Nobuhle and I were singing at some point. And at some points we'll be very tired and just resting a little bit. But yeah, so we'll do our 24 hours trekking, take a day off, and then continue another 24-hour trekking the following day. So yeah, this is just some of my favorite moments in the water. So these are the results that came from the trekking work. So the shape, there's the shape of the lake, and the colors represent different fishes in the water. So this was February and March 2020 before COVID times. And this is where the fishes were. So they're around the locations of the fishes. And as you can see, there's not much difference between February and March, especially in terms of where the fishes are located. And then where they are is usually where the reeds are. So they go closer to the plants. And as I mentioned, that's where they eat. So this was just the first part of our results sections. And then we were stopped due to COVID-19, had to rest a little bit until we could go back and collect some more data. And this was in October and November same year. So when we went back in October, some would say October is still similar to February and March, which is, I agree. But then if you look at November, you see kind of like a pattern. And this is where the fish were located in one very small spot. So I've shown here, calling them the spawning grounds. So this is what they call backwaters. So it's still connected to the lake, but it's a very small area that is very shallow in depth. And then to a point that even our boat, we struggle to drive the boat into that small space. It's about 344 meters, which is very small. And all the fishes that we tagged were in that particular area, which was very interesting for us. And then what we did, we also observed the fishes go splash in the water. So this is known as the spawning behavior of fish. And then from my other chapter, which is the biology, we realized that carp at Groenvlei spawn around November, and this is during that period where they spawn. So we were lucky now to actually get to see the carp spawning and record it for our ecological data as well as the biology. So now in that small section where the carp were located, this we identified as a spawning ground, which I'll tell you why this is important. And by February next year, the fish had already moved back to the lake, and their movement was similar to the ones I've shown. Okay, so just a summary of our major findings and conclusions from this project. So acoustic telemetry was valuable in a way that it showed us that during spawning, carp have a distinctive movement that they do. And they actually move to a certain point and stay there for their spawning period until when it's done and they go back to the lake, which was the following year in our results. Yeah. So that method of tracking helped us see where exactly that location is, and we were able to map it out on a map. And then we also learned fine movement patterns. I mean, there is multiple types of acoustic telemetry. You could do passive tracking, but then with our lake being very small, it was better that we did manual tracking, tracked individuals, and then find their finer movement patterns. And then, which in this case helped us identify where they spawn. And then, if you notice, some of my pictures are from IFSM, which is a non-profit organization around Groenvlei, and these are volunteers who actually go into the lake and collect mostly carp, and then to kind of reduce its numbers. Yeah. So I have a picture that they use bow and arrow method a lot, and they've been using this before we came into the picture with our research. So the reason for this is that Groenvlei is in a conservation priority areas in a nature reserve, which means that species such as carp, which are number 1C on the number our policy, need to be in check in a conservation priority area. So if we have a nature protected area, species such as this need to be checked because their impacts can be very significant. So they've been doing this program of trying to reduce numbers of carp at Groenvlei for a very long time. And then we came in because they were sometimes struggling to get high numbers of carp. So we needed to show them where exactly can they find them, or if we can help them improve their methods. So so far with their program and alongside Cape Nature, which is the management authority there, they've been involving other people, other members of the community as volunteers, and they go into the lake and they actually still do bow fishing and kill a lot of carp. But then now they're targeting areas that we suggested, especially the backwaters. So I saw an article on their Facebook page from August actually, and something interesting about it was that they've been catching quite a lot of carp recently. And they're using even gill nets, and then they use even fight nets and bow fishing. So like it's a whole integrated methods to reduce the number of carp. But then carp, as I mentioned, that it was introduced for food as well. So other people may not like carp, might not like to eat carp. But then with COVID-19 and people being affected a lot by the pandemic, so Gift of the Keepers, which is a charity organization, started feeding this carp to the people, of course cleaned and prepared meals. Yeah, so now this carp that they catch goes to the kitchen, a lot of it currently, which is very good because people have started preparing it differently and people are loving it. I've tried it, I love it too. So yeah. So my colleague mentioned that we went to Belgium, which was our highlights, I think career highlights. So this work was also presented in Belgium, and this is us heading out there to the world. And then this, the building in the background is where I think the conference was. So the place where the conference was, it was beautiful inside out. We were by the coast, and we presented in a session honoring the memory of Olaf. And then Nick started with a very beautiful tribute to Olaf, which was very touching. And then after that, we continued as people who worked with him and on stuff that he was interested in, with a bunch of presentations from there, and that was very much amazing. And then, so we also got opportunity to network at the conference, and some faces that we were seeing again, like Josie South, who was here at SAIAB, and then we got to see her again, which was nice. And Paul and Nick also came to visit us earlier in 2020, and then we got to see them again, which was very nice. And we also got to meet Nick's lab, which they're doing lots of interesting stuff on telemetry, which was interesting for me. And then the other guy on the other side, Sa, was in the local organizing committee of the conference, which for me, I found it interesting because I'm interested in organizing stuff. So yeah. Yeah, so we also went to the Netherlands to see some of the invasive species. And the picture there in the bottom middle is me looking at invasive oyster in the Netherlands. And as you can see, we're on top of it. It was actually so much that you couldn't see any surface. And this is next to the water, so it's overtaken the whole area and it's just the only thing growing there. So it was interesting to see how much they have over there. And yeah, people do eat it, and although it needs some kind of control because it's a lot, and other species can go. So some of the pictures I put here, which is some of my highlights, are looking at the tram there in Belgium, which I thought it looks like a hull train from South Africa. Yeah, the difference is that it drives in the middle of the city while hull train goes under. And then this is Nobuhle and I in a ferry. We were crossing water body, large water body, to go into some parts of the Netherlands, which was cool because when we came back, we didn't take the ferry. We actually went on an underwater tunnel, so that was cool. And then some of those. The other one at the top is at the beach. They had like nice place for kids at the beach like me. Yeah. Yes, that's a summary of my experience there, and I would like to dedicate this presentation to Olaf again for this wonderful opportunity, because it all started with his email. And then me coming down here has been everything following that. And the funding for my master's and the funding to go to Belgium goes to the ICAS bursary and NRF SAIAB and Cape Nature, which was the management authorities where we were working. They did help us a lot. And for field work, you saw some of the pictures, and some people are doing great pictures of them, but I put them there to thank them for their contribution. Thank you so much, everyone.
Thank you very much, Dinah. So I have some questions. Quite a lot of questions. So are you two ready for questions?
No.
Okay, I'll start with a question to Mukhari. Now that we know what makes the competitively dominant over the native one, what potential strategies could we employ in those ecosystems to ethically remove or suppress numbers of the invasive species?
Thank you, Linda, for that question. So there's a group of researchers of science who are doing this amazing work on the distribution of tilapia in South Africa. And I think I will refer you to Dr. Magoro, who's going to be like the amazing person to answer this question for you, because he's already doing distribution, whereas my work was mainly focusing on the functional responses, the biology, the morphology, in order to try and understand how advantageous the invasive species is over the native species, in order to better manage the native gene pool of Mozambique tilapia. So Mandla Magoro is the person for you to, I'll refer you to that person.
Oh, thank you. So I do another question to you?
Okay.
I'll do it. Nile tilapia, this is a question from William. Thank you, William. Nile tilapia adults are hypothesized to feed on eggs and juveniles of Mozambique. With your results and the literature review, are you able to say this could be like that?
Okay.
Nile tilapia, short question, do adult Nile tilapia feed on the eggs and juveniles of Mozambique?
Based on my literature review, based on my literature review, I only reviewed the parts about their hybridization, where they're producing viable offspring which can interbreed with either one of the parent species. I didn't really look much into whether the Nile tilapia is eating the eggs of the Mozambique tilapia, if that's the question.
I think that's the question.
No, I didn't look into that.
When you did your food model, you were sort of telling us that they have quite a broad diet.
Yeah.
Do you think it's feasible?
It's possible. They're eating a diverse set of food items compared in freshwater aquatic system compared to the native one. So it's possible. Even though I haven't looked into that, but it's possible. Yeah. I'll look into it.
Thank you very much. I have another question for you. This one's for you. Anybody out there wanting to ask questions for Dinah? We're persecuting the bushfire a little bit here. But more Dinah questions. Anyway, let's go for one from William. apart from carp, what other species exist with carp in Groenvlei? Are you able to give percentages or presence percentages of your catches?
That's a very good question. So there is 6 more species that are at Groenvlei, and then there is 2 of them are native. It's Stella and I think so. Yeah, Mozambique. Yeah, no, Nobuhle fish is there as well. And then we have introduced fishes. We have carp, we have bass, we have mosquito fish, we have bluegill fish. I think I'm forgetting one more. But yeah, so carp is the main species there, because even bass, people who used to go there catch bass, they're actually complaining that they can't catch it anymore. They even cancelled their competitions because there's no bass around. And the rest of the fishes are just hard to find, but I never actually used maybe the right methods to look for those fishes. So I'll be using bigger to catch for big carp. So I wouldn't be certain how much percentage.
Thank you very much, William, for that question. Okay, another one for the that's a genetics question as well. Let's leave those for
Mantra, yeah.
Okay, this is one I hope you can answer. Hi, Nobuhle. So it doesn't say your name, but thank you for your question. Can I ask how long the Nile tilapia has invaded freshwaters in South Africa, and if there is possibly a biomass estimate of the species?
How long?
How long have they been in South Africa?
So they were initially introduced in the Bongo, and now they are in parts of KwaZulu-Natal and the Western Cape. In terms of the biomass estimates, no, I don't have numbers for that one specifically. But yeah.
Cool. Thank you, Nobuhle. Okay, so another one for you. This question is from Nobuhle. Could you expand a bit on the ecological implications of your findings, particularly with regards to the coexistence of the 2 species? Based on your findings, do you think tilapia would replace Mozambique in resource-limiting environments, or could the 2 coexist? Also, if the 2 species are trophically analogous, do you think there might be other non-trophic impacts the Nile tilapia might exert on the Mozambique tilapia?
Okay. Thank you for that question. So based on my functional response chapter, when looking at the multiple predator effects, as much as Nile tilapia is eating or consumes more prey than the native one, but I found that it's not really affecting the feeding. It does not influence the feeding of the native one. So there is a chance of them coexisting, because Nile tilapia is in the system, regardless whether there's competitors as a native one or not. It's still feeding the way it's supposed to feed, but it's not directly affecting the feeding of the native species. So coexistence definitely in the future might continue happening. But based on my results specifically, the coexistence is there, because from my results, I didn't find that Nile tilapia was reducing the feeding by the native, even though the native was not really inferring any biotic resistance to the feeding of the native, the invasive species. But yeah, coexistence in the future is possible.
That's a good answer. Question for Dinah. Yeah. So, you mentioned that the carp was introduced into Groenvlei. Was there any reason for them to be introduced?
Well, it says in the literature illegally introduced, so I don't think anybody present for something illegal. Yes, I don't think so, because after the impact of carp was here in South Africa 50 years ago, or even more, they actually stopped the introduction of carp. So if somebody introduced it, it was not a good reason.
Thank you. That question was from Lucy. Thank you very much for that question.
Thank you, Melissa.
Okay. William. Oh, this is nice. Not a question anymore. Just a message of confidence towards women empowerment. Thank you, SAIAB. Thank you, William. Yeah. Ladies, these ladies are the future of South African science. So thank you. Let's just see if we've got any more questions. Okay. One more for you. That was an amazing and interesting talk. I agree. I am not sure if I missed it, but I just want to know, what is the way forward for Mozambique tilapia? Will they be conserved, or is more research needed to conserve the genotypic value of the Mozambique tilapia?
The plan is definitely to conserve, which my study was feeding into, but also the current work that is being done, which unfortunately I'm not involved in, but that's also working towards the conservation of Mozambique tilapia in South Africa, because we're slowly losing the population. So we're trying not to, by doing all this different research that you're doing, including my research, which feeds into identifying critical species, native species that needs conservation, like Mozambique tilapia.
Thank you very much, Trace, for that question. Okay, one last one for you. If anybody's got a burning question, type it quickly before we let these ladies go. The question is from Sabella. Having seen that the Nile tilapia is much more successful, what are the negative impacts this could have on the ecosystem?
Thank you, Sabelo. So for a system to function, we need to have all the major players there. In the case that you're losing a certain species, that's not good for the environment. So in my case, we are trying by all means to make sure we're not losing the genetic, the natural native species in South Africa. And that's what my study was aiming to do, to conserve the native. I've just said this in the previous question. So the plan is to reduce the number of the invasive, as much as we're recognizing the economic value that it has in terms of food security, because more people are preferring Nile tilapia for aquaculture because of its fast growth. Although that's the case, we're also trying to actually highlight the impacts it has on ecosystems and also conserve the native gene that's existing. I don't know if I'm answering the question, but I hope I tried.
Okay, that's it for the questions. It's now around 11:30, so I would like to thank very much both our speakers today, Dinah Mukhari, Nobuhle Mpanza. We were very proud of you, and well done, and best of luck with your continuing PhD studies.
Thank you.
Well done.